Hello, everybody. This is Nafrit Sekho, and I welcome you on this platform, The Layers of Life Sciences, where we are discussing various aspects of life sciences in quite depth and detail, layer by layer. We have started with biochemistry, and in that, we are discussing the structure of carbohydrates. In last videos, we have discussed the linear structure of monosaccharides, which are also the basic building blocks of carbohydrates in quite detail. This linear structure of monosaccharides is an absolute truth for all the monosaccharides, which are composed of either 3 carbons or 4 carbons, which means which are either trioses or tetroses. But all those monosaccharides which are composed of 5 carbons or 6 carbons, for them there is a twist in the tail with reference to their structure. So, uh, as in case of every tale, there is always a backstory. So here also, in this tale as well, there is a backstory which is composed of a fact in the chemistry. So before we actually discuss this twist in the tale and its repercussions on the structure of monosaccharides, which are either pentoses or hexoses, before that, let's discuss the backstory which lies in chemistry. So let's start with that. First of all, let me introduce you to the characters in this story. So the first character is an aldehyde. So we know what is aldehyde? Any carbon compound composed of certain number of carbons with the terminal carbon having a carbonyl group, right? Other carbons may just fulfill rest of their valencies with the help of hydrogen. So here 2 hydrogens are required, here 2 hydrogens are required, and here just 1. So this is the carbon involved in the formation of carbonyl group, and this whole molecule is basically an aldehyde. Now, if you will look at this closely, just this part of it, you will realize that it is just like an alkane, right? So this part will be same as alkane. So here also there is a single bond between the 2 carbons, and with it between the second and the third carbon also there is a single bond. Had it been an alkane, then here instead of 2 8s there would have been, to satisfy its valencies it needs 3 8s, right? So to accommodate this carbonyl group over here, one of the 8s have been removed from here, right? So that the only 3 of the carbon's valencies are fulfilled. One by bonding with the second carbon, and another 2 by bonding with 2 hydrogens. So it is one of its valency is not satisfied. Now, whenever such a group is created, which is basically an alkane minus 1 hydrogen, then all such groups are called alkyl groups. So alkyl groups are nothing but they are the derivatives of alkanes. Alkane minus 1 hydrogen is called alkyl group. And in chemistry, these alkyl groups are generally referred as R, uh, the uppercase R, right? So if you will closely visualize this, right now, this is basically a propyl group. To a propyl group, another carbonyl group is joined, so which forms an aldehyde, which is called butanol, right? So over here, this whole of this part is there, which is called butanol, right? Now, it is not about butanol, right? The character which I wanted to introduce you to was an aldehyde, and it can be any aldehyde, not specifically butanol, propanol, or ethanol, or pentanol. No, it can be any aldehyde. So it is very important for me to write the generalized form of the aldehyde. This is the first character of our story. Now, let's come to the second character of our story. The second character of the story is alcohol. So, what is alcohol? Any alkyl group, right? Let it be here, there was like propyl group, here we can take ethyl group, right? When this fulfills its that one valency with the help of single bond oxygen, right, which is further bonded with the hydrogen, then this group, which actually whole of this group is called hydroxyl group. Now, when in this way the valency is fulfilled for a carbon in the alkyl group, then that group is called an alcohol, right? Now, because this is again an alkyl group, over here also we can refer this alkyl group as R. This is also R. This is different kind of R. So to show that distinction, we can either use different colors to differentiate, or we can just say this one just R and this one as R dash. If we will come across any third alkyl group in any other molecule, then we can call it as R dash dash. But another good way is that we call this R as R1, and this another R, which is associated with the hydroxyl group and forming an alcohol, that can be called as, that can be referred as R2. And this alkyl group gets bonded with oxygen and which is further bonded with another hydrogen, then that group is called alcohol. So these are the two major characters. Apart from these two, we will come across a third character, which initially will not have any role to play, but we need to recognize whenever it comes into picture. That third group is derived from this alcohol only. For example, this is simple alcoholic group, right? This is CH3. CH2, it binds to oxygen. Now, this oxygen, if it chooses to bind with hydrogen, then this is simply an alcohol, right? But if this oxygen, instead of binding with hydrogen, just gets bonded with another carbon, and that carbon may fulfill its valency with the help of three hydrogens, like three valencies will be left behind, right, after binding with oxygen to fulfill its tetravalency. So it may just fulfill that valency with three hydrogens, or it may just get associated with just two hydrogens and bonded with another carbon. So this this, if you would say, this is actually same alkyl group. It is CH3, right? So it's just three valencies are fulfilled over here. One hydrogen is lacking again. This is also an alkyl group. This much over here we have referred as R2, right? So if this is R2 to this R2, and oxygen is bonded over here, and to this oxygen, there is another alkyl group bonded, and we need to name it as well, right? So let's just name it as R3. If it is ROH kind of bond, it means it is an alcohol. But if it is ROR kind of bond, then this kind of molecule is called ether. So these are the three major characters of our backstory. Now it's time to see their action. For action, let's come to this diagram over here. So if you will see this structure over here is just the same, right? So this is R1 and this is carbon, and this carbon is bonded with the carbonyl group and a hydrogen atom, right? So this is aldehyde basically. So this is aldehyde, and this over here is an alcohol, ROH group, right? So this is alcohol. So the first action is that whenever an alcohol interacts with any aldehyde, then that alcohol leads to the shift of electrons of one of the bonds of double bond towards the oxygen. Now, when the electrons get shifted to oxygen, the oxygen gets a negative charge and the carbon gets a positive charge, which you can see over here. Now, this shift of electrons comes from a phenomenon in chemistry which is called electromeric effect. So what is electromeric effect? It is when the electrons of one of the bonds of a double bond get shifted towards a more electronegative atom in presence of an agent, then that shift is called electromeric effect. The point to be underlined is in presence of an agent. So the agent over here is the alcohol. This alcohol is causing the shift of electrons of this bond towards the oxygen. The action 2. There is a bond between this hydrogen and this oxygen. This bond over here undergoes a heterolytic cleavage. Heterolytic cleavage is when the bond breaks and the two electrons involved in the formation of that bond after cleavage get shifted to just one of the atoms. If the bond breaks and after that breakage, the electrons involved in the formation of that bond get shifted, one goes to one atom and another goes to another atom, then that cleavage is called homolytic cleavage. But when after cleavage, both the electrons involved in the bond formation just get shifted to one of the atoms, that kind of cleavage is called heterolytic cleavage. So over here, between this oxygen and hydrogen, a heterolytic cleavage takes place and electrons get shifted to oxygen. So oxygen will become negative and we are left with a positive hydrogen because the only electron which it has lost to oxygen. Now, so actually in alcohol as well, now two different species have been created. One, a positive species, and another, a negative species. This OR negative is basically a nucleophile. So it is nucleus loving. There is nothing called nucleus loving. It is basically positive loving because nucleus is positive. So the name is given as nucleophile. And another one is electrophile. Electrophile means electron loving. Electron loving means negative loving because electrons are negative. This was the completion of the action 2. Now, what is the action 3? Action 3 involves the nucleophilic attack by this nucleophilic oxygen over here on this carbon. Once this nucleophilic attack is made by this oxygen on this positive carbon, it succeeds in the bond formation over here. You can see over here. So it succeeds in the bond formation. A new bond is formed between over here, right? A new bond is formed over here. And OR2 just joins, this whole of this group just joins the molecule. Right? After that, we are left with O negative. This O negative then binds with this electrophile. This is H positive is electrophile, means negative loving. So this H, which we have got from alcohol, just joins the oxygen. So you can, first thing which you can see is that this is a nucleophilic attack is happening, and after that nucleophilic attack, an addition reaction is taking place. What is addition reaction? Whatever the components of the attacker were or the agent were, they all get accommodated within the same molecule. They just find their places within the same molecule. The components of the two molecules just form a one molecule now. So this molecule is formed over here. Now let's analyze this molecule very closely. It's very important. So there is a carbon in the center, right? And on one side, it has the same R1 chain. This R1 chain was initially on this carbon only, so it remains at its place, right? And then this hydrogen, which was there, this is also intact. Now, whatever the changes are happening are on this oxygen bond. Earlier, this carbon was bonded with this oxygen with the help of double bond. Now, only single bond exists. And now it is not only just oxygen. This oxygen has further bonded with hydrogen. So on this carbon, now instead of a carbonyl group, there is a hydroxyl group, right? And apart from that, if you will just see this, this is, if you just consider whole of this R1, right, including this carbon, this is R1O R2. So this is the formation of an ether group. So on the same carbon, there are two functional groups. One is the ether group and another one is the alcoholic group. This carbon, this is just a rearrangement, right? This was, we have just kept the same kind of system till this is just a rearrangement. H has been shifted. So if you will just look at this, it is one and the same thing. Now this carbon, or you can say this carbon, earlier this was not a chiral carbon. Now it is fulfilling all its valencies with help of four different groups. Here R1, here OR, here H, and here OH. So now this is a newly created chiral carbon, number one. This carbon over here is called hemiacetal carbon because the structure which has been formed, this molecule is called hemiacetal. This carbon will also be not known as anomeric carbon. So this is basically the backstory. Now we need to move forward to understand original tale now, right? So let's see what is the twist in the tale. So if we just see this structure, this is the linear structure of what? There is a terminal carbon, there is a carbonyl group on that terminal carbon. It means certainly this is aldehyde, right? This is aldehyde, done. And on rest of the carbons, there are so many hydroxyl groups. So this is polyhydroxyaldehyde, right? So polyhydroxyaldehyde is also known as aldose, right? This is also known as aldose. Now, which kind of aldose it is? 1, 2, 3, 4, 5, 6. There are 6 carbons. So it is aldohexose. This is aldohexose. Now, there are 16 types of aldohexose, right? As we have done in the previous videos. Now, out of those 16 types of aldohexose, which kind of aldohexose it is that? It is, let's see the code. So if you can see, you can find that the code is for the right-hand side. It is 245. It depicts the code for glucose, right? So it is basically a D-glucose. There is a carbonyl group, which is the aldehydic group, and there are so many alcoholic groups, aldehydic as well as alcoholic. Everything is within the same molecule. When we were doing the backstory, we discussed the reaction between two different molecules. That's why that reaction was intermolecular. But now, when everything is available within the same molecule, we don't need another molecule. So now, all the attacks and all the actions will take place within the same molecule. And when each and every action will take place within the same molecule, in that case, the cyclization of this linear structure will take place. How this cyclization takes place? In that cyclization, uh, either the hydroxyl group on carbon number 4 or the hydroxyl group on carbon number 5 will be involved, which means that this alcohol on the carbon number 4 or this alcohol on carbon number 5 will induce the electromeric effect on this carbonyl group, and the electrons will get shifted to this oxygen. So this was the action number 1, right? So let's see how this happens. So this is again the same linear structure, but this time it has been given the proper form. It is just another version of the linear structure which we have seen in the last slide. So this is carbon number 1 carrying the carbonyl group, right? And then the carbon number 2, 3, 4, 5. and carbon number 6. Each one of them are just having their hydroxyl groups and the hydrogen groups, right? This is carbon number 6. Now, as we have discussed in the last slide, either the hydroxyl group of carbon number 5 or the hydroxyl group of carbon number 4 is at the appropriate distance to induce the electromeric effect on this carbonyl group. Mostly the hydroxyl group on carbon number 5 is the one which induces this effect. In the rare cases, yes, this happens that even this hydroxyl group on carbon number 4 may induce this electromeric effect. So this is the case when we need to discuss that how the hydroxyl group on carbon number 5 acts on this carbon number 1. So this alcoholic group comes close to this aldehydic group, interacts with it, and leads to the electromeric effect over here. The electrons of this bond shift over here. This gets the negative charge and this gets the positive charge. This action number 1 is done. Now, action number 2. Action number 2 is that this alcoholic group over here undergoes a hydrolytic cleavage. H positive and O negative is created. Now, this nucleophile leads to a nucleophilic attack on this positive carbon over here and succeeds in forming a bond. Once this bond is formed, then we are left with this H positive, which is electrophilic. Now, this H is free H and it just joins this oxygen over here and OH is formed. So now once whole of this thing is done, then this structure is formed, right? A bond with this oxygen of the alcohol, which was actually bonded with carbon number 5. This hydrogen comes over here. Right now, as we discussed in the first slide earlier, this carbon was not chiral but as now it is connected to 4 different groups. Now this is chiral. Now, because this is chiral, so whenever the chiral carbon is there, as we have discussed in last 2 videos, whenever there is any chiral carbon, it leads to stereoisomerism, right? Now, if stereoisomerism is there, there must be some stereoisomers. So over here also, there are 2 types of stereoisomers which are formed. One is called alpha and another is called beta stereoisomer. It is alpha if hydroxyl group is pointed downwards. But if this hydroxyl group is pointed upwards, like in over here, then this group over here is basically a beta molecule. We have discussed the mechanism of cyclization in the case of aldohexose, and that specific case was that of a glucose. Now, we need to discuss few general things. So these kind of reactions where an alcoholic group is attacking a carbonyl group on a terminal carbon, this kind of reactions are possible in 2 kinds of groups. Ones are aldohexoses and another are aldopentoses, right? These are the 2 aldoses where this is possible. Aldotrioses and tetroses, just stay out of it. So in the case of aldohexoses, generally what happens that the hydroxyl group on carbon number 5 leads to the electromeric effect on the carbon number 1 and attacks it and further attacks it in a nucleophilic attack. So it is generally it is 5-1 carbon C5 and carbon C1 interaction. While when we come to the aldopentoses, generally, so if we draw an aldopentose, for example, let's draw ribose, that is the most important aldopentose. So in case of ribose, this is made up of 5 carbons, the terminal carbon having double bond O and the 5th carbon CH2OH, and the central carbons have the code 234. Right, so on second also, on third also, and on fourth also. On all of them, hydroxyl group is on the right hand side, And if it is on the right hand side, then on the other side there are the hydrogens. So this is the basic ribose structure. Over here, this hydroxyl group on carbon number 4 conducts a nucleophilic attack on this carbon number 1, right? So that interaction is basically between a group on carbon number 4 and carbon number 1. So if it is the interaction between the groups on carbon number 5 and carbon number 1, then it means in the ring formation, how many carbons will be involved? 5 carbons. So here, always 5 carbons will be involved. And over here, because general interaction is between carbon number 4 and carbon number 1, so over here, 4 carbons are involved. So whenever the 5 carbons are involved, in that case, the hexagonal structure is formed. In that hexagonal structure, so if let's draw a hexagonal structure. So in that hexagonal structure, what is the common thing? That the 5 angles are occupied by the 5 carbons. So we start from this right hand side. And from the right hand side, if we start writing, we start making showing the carbon number 1. Then followed by carbon number 2, followed by carbon number 3, followed by carbon number 4, followed by carbon number 5 over here, right? And on this carbon number 5, there is carbon number 6, which is attached as CH2OH, right? So this is done. And on the 6th angle, we actually draw this oxygen, which actually attacks this carbon, right? Forms a bond with it. So whenever the 5 carbons are attached, then 5 plus 1, a 6-angled hexagonal structure is formed. And whenever 4 carbons are involved, in that case, 4 plus 1, 5-angled pentagonal structure is formed. So the skeleton which is formed is pentagonal in that case. So there is carbon number 1, carbon number 2, further bonded to carbon number 3, further bonded to carbon number 4 over here. And this carbon number 4 is actually the carbon, is actually bound to the oxygen, which attacks this carbon in a nucleophilic attack. General hexagonal and pentagonal structures. These hexagonal and pentagonal structures, which are created after the cyclization, depending upon how many number of carbons are involved in that cyclization, so they are formed. This hexagonal structure actually resembles an organic molecule, which is called pyrene. And this resembles an organic molecule, which is called furan. So because it resembles the pyrene, whenever a cyclic structure is formed by a monosaccharide, that is called pyranose ring. While if a pentagonal structure is formed by a monosaccharide in the process of cyclization, then the ring which is formed is called furanose ring. Now, so from here, from the word pyranose and furanose, actually we get to know that how many carbons are involved in the cyclization. And from here only we get to know that hydroxyl group on which carbon is attacking the carbonyl group present on the carbon number 1. This is the first part. This is the linear structure, which has been converted into a cyclic structure. We know that this is glucose. This is basically D-glucose. How do we know that? We know that because there is aldehyde group. So this is, and this is, and there are 6 carbons. So this is aldohexose. Which aldohexose it is? It is the aldohexose with the code 245. 245 is the code for glucose, right? So we know that this is the D-glucose. Why D-glucose? Because on the carbon number 5, which is the farthest chiral carbon from this carbonyl group, the OH is directed towards the right-hand side of the chain. So this is D-glucose. So if we are cyclizing this linear molecule, it means we are creating a pyranose ring out of which molecule? It is D-glucose. So while writing it, we don't write whole, the name of whole of the molecule, we write D-gluco. D-gluco. So how to create that D-gluco in the cyclization? So there is a rule over here. Whatever is on the left hand side of the chain, whichever groups are on the left hand side of the chain, they will point upwards on this ring, while all those which are on the right hand side of the chain, they will be pointed downwards. So on the carbon number 1, this is a newly created chiral carbon, right? So therefore the H and OH, this is the anomeric carbon now, right? So this will be our stereoisomer, right? So over here, if I have written OH upwards, it means the structure which we are forming is basically beta D-glucopyranose. Beta because it is directing upwards, D-gluco because now this cyclic structure is that of glucose molecule, pyranose because carbon number 5 in the action on the carbon number 1, right? So this is that like 5 carbons are involved. So this gives us the name, gives us the structure, right? So on the carbon number 2, we know that over here, this is the carbon number 2. On carbon number 2, needs to be directed upwards, right? And OH needs to be directed downwards because it is on the right hand side of the chain. On the carbon number 3, OH is on the left hand side, so upwards, uh, and H is on the right hand side, so it should be directed downwards. Over here, CH4. So over here, H should be directed upwards, and over here, it should be downwards, right? On the carbon number 5, carbon number 5 is no longer with OH over here. So it is just O. This O is already written over here. The another group which is bonded with it is CH2OH. It is always written upwards in the case of D-glucose. And the other group is H. H is always written downwards. So this is the cyclization of basically D-glucose. This is the cyclization of D-glucose. After cyclization of D-glucose, the structure which we have just drawn over here is beta D-glucopyranose. This is one thing, and this is the general structure. This is how the aldohexoses generally form this pyranose kind of ring. Now, if we come to the other part, which is aldopentoses, over here, we know that carbon number 1 forms the oxygen bond. Now, this carbon number 1 can be associated with OH. It is associated with OH and H. This OH may be directed downwards or upwards. Here we have drawn it downwards. So this is basically alpha form, right? So this is the alpha form done. Okay, which molecule we are actually converting into this cyclic structure? So we can get, so this is basically aldopentose. Which aldopentose is? It is the aldopentose with the code 234. 234 is the code for ribose. So it is ribo. D-ribo because on the 4th carbon, OH is on the right hand side. So it is alpha D ribofuranose, right? Alpha D ribofuranose. So to just copy the structure into the cyclic form, the rules are same. Whatever is directed on the left hand side should on the ring, it should be drawn upwards, and everything which is on the right hand side should be drawn downwards. So if we go to, this is the carbon number 1 over here, let's we go to carbon number 2. This is carbon number 2 over here, right? So on the carbon number 2, H is on the left, so upwards, and OH is on the right, so downwards. H is on the left, upwards, OH is on the right, upwards. So on the 4th carbon, if we check, there is no OH left. O is already participated in this bond formation with the carbon number 1. So this is the ether group formed. No OH, but on this carbon number 4, CH2OH is there, and this H is there. This CH2OH in the case of D forms is always shown upwards, right? And H is shown down. So this is the cyclic structure of alpha D ribofuranose. So these are the general structures. So the first difference is which carbon is attacking which one and how many carbons are involved. Here 5, here 4, because there's the difference in the number of carbons involved in the cyclization. Two different kinds of structures are possible. There can be so those structures can be hexagonal, it can be hexagonal, or it can be pentagonal. If it is hexagonal, then that ring will be called pyranose ring, and if it is pentagonal, then that ring will be called furanose ring. And whatever monosaccharide is forming that pyranose or furanose ring, then the prefix of that monosaccharide is written along with which enantiomer it is, whether it is D or L, the full name is written. And then finally, in the front, which stereoisomer of that cyclic structure has been formed, that is mentioned in the front, whether it is alpha or beta. These are the general things. Now, for example, even if we are talking just about glucose, glucose tends to exist in basically 5 forms. First form is this linear form. Second form is this form, beta-D-glucopyranose. Second, it is the first abundant, right? Then second abundant, it will be alpha-D-glucopyranose, right? So that second form which can be formed is alpha-D-glucopyranose. Now, so there are 3 forms in front of us, right? Which can be. Apart from that, sometimes what happens, instead of this oxygen on carbon number 5, the oxygen which gets involved in the ring formation is this oxygen which is there on the carbon number 4. If this oxygen takes part in the nucleophilic attack, then in that case, only 4 carbons will be involved in the formation of the ring. So I have told you, whenever the 4 carbons are involved in the formation of ring, then in that case, always and always furanose ring will be formed, a pentagonal ring will be formed. Now, if the pentagonal ring will be formed, it means beta glucose can exist in the form of furanose form also. So another form can be beta D glucofuranose. This happens rarely, right? Generally, only the hydroxyl group present on carbon number 5 attacks the carbonyl group on carbon number 1. But this also happens, right? It's not like that it never happens, that if this is aldohexose, it can never exist in case of furanose. No, it is not. It is always present, but in low amount. So beta D glucofuranose will be the 4th form. Alpha D glucofuranose will be the 5th form. While in the case of aldopentoses, the most abundant are the furanose rings. They can be either alpha or beta furanose rings, right? But sometimes this hydroxyl group present on carbon number 5 interacts with this carbonyl group on the carbon number 1. If a ring is formed in that way, it means in that case 5 carbons are involved in that ring formation. If the 5 carbons are involved in any ring formation, in that case, a hexagonal structure is formed. And that hexagonal structure is called pyranose. So it means aldopentoses can also form the alpha or beta pyranose rings, right? So they can also exist in all the 5 forms. So whether they are aldohexoses or aldopentoses, both of them can exist in all the 5 forms. But for aldohexoses, most common forms are the pyranose rings, while for aldopendoses, the most common forms are the furanose rings. So with this, we have finished with the cyclic structure of the aldoses. Today we have done the cyclic structure of aldoses. In the next session, we will do the cyclic structure of ketoses. It will just be the revision of whatever we have done today. And after that, we will be discussing the analytical questions which pertain to this part of structure of monosaccharides. So I look forward to see you in the next session. If you have any questions, any doubts, you are always welcome to ask in the comment section. So till then, till the next time, goodbye. Take good care of yourself.